thank you, Anwen, as well, for that really kind introduction. It is great to see everybody. I've been given a brief to talk about my own project, but also just to talk a little bit about research in ovarian cancer. So what I thought we might do this morning, with your permission, is actually look at ovarian cancer from the perspective of what we've done over the last 20 years in terms of research and where we've got to. Then focus a little bit about our project in terms of the present and what we've just been doing more recently, because hopefully by then I'll show you how it fits into the bigger picture. And then I wanted to end with a few slides just showing you where I think we are going and some of the things that are on the horizon, really to try and set the scene of what research has achieved and what research is going to achieve. I make no apology for some of my slides being quite technical, and I want to show you some very technical figures because I really want to try and show you some of the complexities that are involved in doing this kind of work. I hope the message still comes through, so please don't worry too much about the detail, but it's really to try and give that impression of some of the challenges that we face when we're trying to do this kind of work and how it fits into the bigger picture. I've spoken at several of these kind of meetings before, and obviously I see patients in clinic on a regular basis, and one of the things that always comes through when we talk generally about ovarian cancer is these three questions. And patients will often ask me, why can't we all get the same level of treatment in the UK as other people get? Anwen has just referred to the fact that there are inequalities in care in the UK, and I'm going to highlight that in a minute because I think that's really important, and to some extent remains a bit of an elephant in the room that we haven't yet dealt with but do need to deal with desperately. Because if we could improve standards of care for everybody to the best levels that are already being achieved, just that single intervention would make a huge difference to outcomes for patients in the UK. The second thing that is always raised is, can we have better treatments? And I think by better treatments, people want treatments that are going to be effective and help patients to either be cured of the disease or at least to live longer with the disease, but most importantly to maintain quality of life through that. And how we choose the right treatment for the right patient at the right time remains a real challenge, and that's sort of where I'm going to get to with what our project has been trying to achieve. And then the last one is always the question of, could my disease have been picked up earlier or could it have been prevented? And we know that ovarian cancer tends to present late at an advanced stage when it's already spread outside of the ovary and the pelvis, and that obviously makes treatment more difficult. And so the question is, can we do better with that? So I'm going to try and focus a little bit on those three questions as we go through that, but hopefully show you, and my sort of overriding message in the next 40 minutes is the fact that if we go back and try and understand more about the disease, it provides the key to trying to unlock the answers to some of those questions. So I'm going to try and convince you that it's really important that we do the science behind this, because understanding the biology of the disease and how it fits and how it all works together actually enables us to try and answer some of these really important clinical questions. So let's just start with this problem of inequality that we have in the UK. And as I say, I think this is the one area that we have still failed to address. This graph here is data that was taken from something called the Ovarian Cancer Audit Feasibility Pilot Project, which is a bit of a mouthful, but it was a national audit of how we were doing in the UK. And it looked at what we call registry data, so it looked at the data that's collected nationally about how patients are being treated. The 19 bars across that graph are the 19 cancer alliances in England. So these are the geographical areas which are sort of the administrative management of cancer alliances. So for instance, Greater Manchester is one cancer alliance. And what the different colours show you is how patients with advanced ovarian cancer were treated in those 19 different areas. The red bar at the bottom is having surgery followed by chemotherapy. The orange bar is chemotherapy, then surgery. Green is chemotherapy alone, light blue is surgery only, and the purple bar at the top is no therapy at all. And what I want you to get from this is the huge differences that exist across the country. So if you look at patients who have any combination of surgery and chemotherapy, which in many ways is probably the ideal treatment, then it can be as low as 25% of patients receiving that combination of chemotherapy, or in some places it's over 50%. So there's a doubling in the difference between those two treatment regimens, and we can't explain that just by patient mix alone. So something is happening between these different areas leading to those different treatments, and we need to somehow unpick that and move forward to a position where we've got the best care for all across the country. So that's our number one challenge to try and deal with. But I think yes, so those are, there are 2 or 3 different timings, but it's about 2016 to 2018. Yeah, so it's not that old, okay? Yeah, relatively recent data. Okay, so let's think a little bit about if we could try and develop a better understanding of the disease, would that lead us to better treatments and focus on the disease in a different way? When I started in gynaecology in the year about 2000, we were very didactic. The gynaecology included 4 distinct diseases. There was cancer of the ovary, cancer of the endometrium, cancer of the cervix, and cancer of the vulva. There were 4 different diseases. They were managed very differently, and they remained in their little silos and their little boxes. But by and large, we considered each one as being a single disease. I think the first realization that we made was actually they're not all the different diseases, and that there are some similarities. Particularly cervix and vulva cancer are predominantly HPV related. They're caused by a single virus. And we've been able to, through the vaccination program and the screening program, really make big advances into how patients are managed and how patients survive with those 2 different diseases. So over the next 50 years, we're likely to see cervix cancer be eradicated from the UK. So that's a real achievement, and it came through an understanding of the biology of the disease. Endometrium and ovarian cancer, what we began to realize again over probably 2000 to 2010, that in many ways there was overlaps between the 2 and similarities between some types and differences between different types. So treating them just as ovary and just as endometrium is not necessarily the best way of going about it. And instead, we need a different way of classifying those diseases and looking at them and accepting that there are differences within ovary and differences within endometrium and overlaps between the 2. Thinking about ovarian cancer specifically, again going back to say 2000, this is the sort of diagram that was in my textbook in the year 2000 that said this is what ovarian cancer is. We treated it essentially as 3 distinct types of ovarian cancer. There were germ cell tumours, which are these very rare cancers that can arise from the eggs within the ovary. And they're a really unusual set of tumours because as they develop, they often develop and grow into different tissues. So sometimes the tumours have different tissues within them, sometimes skin and hair and other things, but very odd tumours. On the other side, there were these relatively rare tumours called sex cord stromal tumours. These arise from the hormone-producing cells in the ovary. Again, very rare, relatively straightforward to treat because we often treat them with surgery, but can lead to problems. But then the big group was thought to be this single group of what we call epithelial tumours. And there were always thought to be different subtypes within that, but they were very much characterised as being the same tumour. So we treated all epithelial cancers as one entity and just treated them exactly the same way. But then 2 things started to happen that really shifted that paradigm and made us think very differently about them. The first was pathology, and the second was genomic sequencing. So in terms of pathology, this work that I'm going to show you here was absolutely revolutionary, and this occurred in the late sort of 2000 to 2010 sort of era. This paper actually came out in 2010. And this goes back to very standard, but very careful and very well done pathology, and largely done by a chap called Bob Kerman in the States, but also by Chris Crum, who was another pathologist in the States. And they started looking, not necessarily at the ovary, but they started looking at the fallopian tube, which of course is right next to the ovary. And when they looked at the fallopian tube, they saw this. So this picture here on the left is a picture of a fallopian tube down a microscope. And you will see the lining of the fallopian tube is here, of this kind of single layer of nice cells that are nicely ordered along next to each other. But on this little bit of the fallopian tube, you can probably make out the cells have got clustered. They're all clustered together and stuck together. And there's more of them. There's a kind of multi-layering. When they stained those areas to look for a particular protein called the p53 protein, which we'll talk about in a minute, and p53 is a key protein in how cancers develop, p53 on this particular stain shows brown. Guess what? They saw that this area had p53 staining in it. What that tells us is this is a precancerous lesion. So they began to find a precancer lesion in the fallopian tube. And to cut a long story short, what they've identified is this is the precancerous lesion that leads to a lot of ovarian cancer. So in fact, a lot of our ovarian cancers that we thought arose in the ovary didn't arise in the ovary at all. They actually arise in the fallopian tube. And that's begun to give us some really important keys about how we might start to attack this cancer earlier, because instead of worrying about the ovary, perhaps we need to think more about the fallopian tube. So there's been a real shift in our thinking since this paper and others were published about 10 years ago in how we think about the origins of ovarian cancer. And don't tell Anwin, but she might need to change the charity's name to Target Fallopian Tube Cancer instead of Target Ovarian Cancer. All right. The second thing that started to happen in 2000 was we were able to start sequencing the genome. So many of you remember in 2000, there was a lot of news about it was the first time that we had sequenced the DNA of the human genome. So we were able to read the genetic code from chromosome 1 right up to chromosome 23, all 3 billion base pairs of the genome, okay? That's only 23 years ago. It's amazing now how quickly that field has moved on. We can now sequence the whole genome for under £600 per patient. Now that sounds quite a lot, but in comparison to the whole effort that went in less than 20 years ago to sequencing one, that's a technology that we now have at our fingertips and can make use of. But it comes at a cost, all right? And the cost is it's very difficult to analyze the genome because it is so big. I'm going to explain why we might want to do it in a minute, but just bear with me about how challenging it is. This is a part of the human genome. All right, so you can see it's made of letters A, C, G, and T. Those represent the individual bases, the individual codes that give us our instruction book in every cell in our body. That's written at size 4 font. That's not the whole human genome. In fact, it takes more than 2 pages. It actually takes more than a book. This is your human genome written at size 4 font for every single cell in your body. Okay, so you can imagine how big that is and how much data and you achieve as soon as you do a single human genome sequencing. And how you make sense of that remains one of our biggest challenges today. We generate a huge amount of data and don't always know exactly how to analyze it. Why is that important? Because it begins to tell us what the genetic abnormalities are in the cancers that we're dealing with. So just taking one step back for a second and thinking about what cancer is a disease of your DNA. Cancers occur when a cell accumulates sufficient numbers of mistakes, it's not one mistake, it's usually several, in different genes that allow the cell to then grow out of control. And once it starts growing out of control, the body can't control that growth anymore, and the tumour keeps getting bigger and then leads to all the problems that come with that cancer. So it's a disease of your DNA. So being able to read the genome should allow us to identify what those mistakes are and then target them in different ways. To some degree, that's been very successful. So what we've been able to do is take this model that I showed you a few minutes ago and actually make it, I'm afraid, more complex, but it now starts to look like this. So instead of having a single group called epithelial cancers, we've now broken them into at least 4 very different and very distinct groups. High-grade serous, which is the commonest, we now think comes from the fallopian tube. And the key gene that leads to that is P53. So a mutation in P53 will lead to a high-grade serous cancer. Low-grade serous cancer, which Anwin referred to, which Charlie Gourley's been working on in Edinburgh, is now a completely different disease from high-grade serous. It's an unfortunate terminology we've ended up with, but it is a very different disease. It's driven by mutations in a totally different pathway called KRAS and BRAF. Quite where it comes from is still a little bit debated. Whether it comes from the fallopian tube or from the ovary, we're not sure. So I've left a question mark there. But we know a lot more about this disease now and recognise that it's different from this one. Mucinous tumours, which have always been fairly rare, we began to realise also have KRAS BRAF mutations, but we also began to realise a lot of those come from the bowel. Okay, so a lot of what we previously called ovarian cancer is now bowel cancer. Okay, so that's another group that's caused a lot of confusion. And then finally, endometrioid and clear cell cancers, a lot of those are coming from endometriosis. So they're not really arising from the ovary either, and a lot of them will have mutations in P10. So the reason I show you this is you begin to realise now we're not dealing with one cancer, we're dealing with 4 very distinct cancers. And the differences between this and this are as big as the differences between this and, say, breast cancer or lung cancer. Okay? They are totally distinct and different entities. Can we take it any further? Well, yes, we can. So I've talked about high-grade serous cancer as being driven by P53. So all high-grade serous cancer has a mutation in its P53 gene. But if you look through the genome, you can find other mutations in other genes, what we might call secondary mutations. And when you try to break it down that way, you get a pie chart that looks like this. So this is high-grade serous, broken down now by its secondary mutations. All of these cancers have P53 mutations, but they've also got a bunch of other things. And some of the names you will recognize, like BRCA1 and BRCA2. Some you won't recognize, like RB1 or CCNE1, but it doesn't matter. They're broken into different groups. So at this point, you're beginning to say, well, what on earth does this matter? Why do we need to break this down and keep reclassifying? Well, this matters because we've then begun to realize that about half of those have a distinct behavior, and they behave in a fairly similar and uniform way, which I'll describe in a second. And the other half behave in a very different way. The group in blue, I'm just going to call them HR defective. I'll explain what HR is in a second, but they're called HR defective. The orange ones are HR competent. Why does that matter? Because a drug that was being developed in, say, since 1980 actually, but first hit trials in humans in 2011, called PARP inhibitors. PARP inhibitors, we realized in about 2013, were very sensitive in the HR defective group, but not in the HR competent group. And that led to us doing clinical trials of PARP inhibitors in that group on that side, the blue tumours, which showed that they are very, very effective at preventing cancers coming back in that particular group. So understanding that biology and unpicking that biology has allowed us to identify a particular group where a specific treatment is more effective. Unfortunately, if you've got an orange tumour, PARP inhibitors are much less effective. So this is where the understanding of biology and genetics and the genomics of the cancer has allowed us to actually get to the point where we can target treatments specifically to specific groups. Unfortunately, as with all these things, life gets more complicated. So we've now split it into several different groups. And what I want to show you with this slide is it's more complicated than that, and it's actually just a continuum and a spectrum of different diseases. So this is work that came out of a UK collaboration where lots of us put tumours together, and then James Brenton in Cambridge led this work to look at the genomics of 575, I think it is, high-grade serous ovarian cancers. And he sequenced each one of them, beginning to end, so all that sequencing data, and then tried to put them together into patterns and signatures. And what he's done here is he's arrayed the tumours by how the patients did. So patients who died very early are over on the left-hand side. Patients who survived a long time are over on the right-hand side. Each one of the vertical lines represents their genomic patterns. And he's summarised it here in this bottom graph. And you can see the trends of how these signatures work from left to right. So it's clear that there isn't a single group that does very well and a single group that does very badly. There's actually just a spectrum from left to right, determined by your particular genome. So life gets more and more complicated as the more and more detail you look at, but it's really difficult for us to pick out individual groups any further than that. All of that, I think, is one way of explaining why we've so far been really unsuccessful with screening. So the question that I'm always asked is there not a screening test for ovarian cancer? Can we not screen for this? Can we not pick it up much earlier than we do now? We've tried, and we tried in a study called the UKC Tox study, which a lot of you will have heard about, where 200,000 women volunteered to take part in screening or not screening. So half were randomised to screening, half were randomised to not screening. And they were followed up for, I think it was about 12 years altogether. The screening was looking with ultrasound and CA125 on an annual basis. And this is the graph that came out of the study. And the way to look at this is there are 3 lines, because in fact there were 2 screening modalities in there. This is the proportion of patients who have died of ovarian cancer as time goes on, okay? And you'll see that the 3 lines are exactly the same. So it doesn't matter whether you had screening or not screening, your chances of getting an ovarian cancer and dying of it, unfortunately, were exactly the same in all the groups. So screening doesn't work using that technology. But of course, we were looking at the ovary, and what I've just tried to explain to you is we were looking in the wrong place. Really, what we should probably have been doing is looking at the fallopian tube. Now, that's not a criticism of the study design. It was the best we had at the time. But I think in retrospect, it explains why that screening does not work. And the message really is you need to understand the disease and get right back to these basic questions of what you're looking for before you start trying to look for it. So maybe we are getting to the point now where we can think about screening again, and I'll come back to that right at the end. Okay, but I think that explains why it didn't work so far. Good. So, having got that far, I think we have learned ovarian cancer is a complex set of distinct diseases. Even with the subtypes that we knew about, there's real heterogeneity, and that's why we've not managed to develop a screening program before. But we have used the knowledge to get new treatments, and they are working really well. Although I'll come back to that elephant in the room, we're still not using the treatments we have as well as we could. So that perhaps leads us on nicely to where we picked up and our research project. So thinking about this, how can we use our conventional treatments better? So we know we've got lots of particularly good chemotherapy drugs like carboplatin that does work very well for some, but doesn't work very well for others. And more importantly, as time goes on, it may not work. So when patients relapse with their disease, it might not work as well second time or even third time. And having tests that would predict whether a patient is going to respond to their treatment or not would be really useful, because it would help patients to decide whether they're going to have chemotherapy at a particular time or not. How much good is this going to do me? And if we could tell them you're very likely to respond, or unfortunately you're not very likely to respond, they may be able to use that information to guide whether they decide to have treatment or not. So we decided to take a slightly different angle than just using genomics, and I'll explain why in a second. And we wanted to focus on this concept. Now, I've already told you that DNA is the cause of cancer, and it's accumulating all of these mistakes in your DNA that allows a cell to become cancerous. So just asking one question back from that, how does that DNA damage occur? Or more importantly, how does it persist? As we sit here in this room, all of our DNA is actually being damaged all of the time. And if in the break you go outside for a cigarette or even stand in the sunshine, the rate of that will go up. So our DNA is being damaged all of the time, but it's not a problem because we repair it. And we repair it using one of 5 different mechanisms. 3 are used when there's just a nick in one strand of the DNA. Remember, DNA is a double strand. It has two sort of backbones to it. If one strand is broken, we use one of these 3 pathways. If both strands are broken, we use these 2 methods over here. You might, if you're really clever, have worked out already homologous recombination is HR. That's what I was referring to earlier when I talked about HR proficient and competent cells, okay? So cells that have a defectiveness in their homologous recombination pathway, in other words, they can't repair their double-strand breaks, they're the ones that are really sensitive to PARP inhibitors. But our hypothesis was that cancer cells, to develop into a cancer cell, must have a defect in at least one of these different pathways. Otherwise, the cell would repair its DNA damage, but clearly it's not, it's accumulating damage. So we hypothesized that number 1, every tumor would have a defect in at least one of those pathways. And secondly, we hypothesized that the different pattern of those aberrations, whichever one of these was broken, would help determine whether patients were going to respond to chemotherapy or not. So instead of focusing on the DNA, we focused on how DNA is repaired and these 5 pathways. And the reason we chose to look at it from a functional perspective and looking at the pathway rather than just the genomics was kind of the analogy of when you take your car in to be fixed to the garage if there's a problem. The mechanic, the first thing they do is not strip your car down into every single component part, lay it out across the garage floor, and then go from left to right trying to work out where the problem is, okay? Which is actually what we do when we're doing sequencing. We're looking from left to right and just trying to see where the problems are. Instead, what they do is they test drive it or test it in some way to see what is actually broken and then focus in on that problem instead. So this is the drivetrain from an MGB Mark 1 Roadster. I used to own one of these a long, long time ago, and I have taken it down to almost that level of breakdown, and it's a pretty complex thing to deal with. So instead of doing that, let's look at things from a functional perspective and see, can we road test our cells to try and see what they can and can't do, and then use that to determine which of these 5 pathways is broken. There was another reason not to use genomics. The problem is all the genes overlap. So we know which genes are involved in some of these processes, but the pathway is so complex we thought we couldn't work that one out. So we'd use a much simpler technique instead. So this is the model that we used in our research. So this is the project that was funded by Target, and we started this pre-pandemic. We did pause a little bit because, as I'm going to explain, it uses patient samples, and we had a problem getting into theatres to collect samples. Even though I work in theatre, it was actually really difficult to accumulate samples during the pandemic itself. So there was a natural pause, and clearly a lot of our labs closed down for a few months as well. But the model we decided to build was to use real patient samples. These aren't cell lines, these aren't animal models, these are ovarian cancer cells that come from our own patients. We take them from the time of surgery, we break them into a single cell suspension, so we take a piece of tumour and we just break it up into tiny bits, and then we grow it in a dish in the laboratory. So if you come over to St Mary's, we have our operating theatres on level 2. If you come up to level 5, we've got a laboratory with an incubator like this, which actually has live ovarian cancer cells in it from the patients who are actually recovering on the ward downstairs. Those cells can grow, they grow for quite a period of time and they expand. So as they expand, we end up with enough cells that we can then do experiments on. So we were able to collect all the clinical data from our patients. We know what's happened to them because we're looking after them. And at the same time, we could develop some assays here to test those 5 different DNA repair pathways and then integrate the results of those with how the patients did to try and see whether we can predict the outcome using the assays that we developed. Okay, so that was kind of our schematic that we used for our project. I want to now just show you what those assays look like. I'm not going to go into the technical details, but it took us, it's fair to say, it took us about 5 years to actually develop and perfect the assays that we wanted to use. So each of them involves the same principle, taking those live cells, in some way inducing DNA damage. So we need to break the DNA artificially in the lab, and then we need to see how the cell repairs itself and measure that to see whether it can repair the damage that we've put in. So as an example, this is how we measure homologous recombination, one of those double-strand pathways. You've got cells on the right, cells on the left. So these are real ovarian cancer cells from patients. The red is the whole of the nucleus that you can see there. If we've induced DNA damage, we've split the DNA, we've cleaved it, we've made it break, and then we've watched to see whether it can repair. If it repairs, because of the way we've set it up, it fluoresces green every time a bit of DNA repairs. So on the left-hand side, you can see little green dots, which is where the DNA is being repaired. On the right-hand side, there are essentially no green dots. That's a cell that can't repair by homologous recombination, so that's a way of testing between the two. For non-homologous end joining, we use a slightly different assay. Here we use a piece of bacterial DNA, which normally tries to form a ring. We've split it. We put the piece of bacterial DNA into the cell and watch whether the cell can stitch it back. If it can stitch it back and make a ring, it goes green, okay? If it doesn't go green, it's because it hasn't managed to stitch that piece of DNA back together again. So again, on the top row, you can see green cells throughout the experiment. On the bottom row, the 2 right-hand ones are black because there's no green, there's no repair going on. For 2 of the single-strand assays, so this is nucleotide excision repair and basic excision repair, so this is 2 of the ways you fix your single-strand breaks. We use something called a comet assay. In a comet assay, we destroy the DNA and then watch to see how it builds it back up again. And then we get the DNA and we stretch it out across a slide. If the DNA is all repaired, then it all sticks together because it's big bits of DNA. But if we try and draw it out and it forms a comet tail, then we can see there are little bits of DNA that haven't been repaired. So we can measure how much DNA repair has taken place in either of the 2 scenarios, depending on what we've seen. And then finally, we measure mismatch repair using a panel of genes that represent that very well. So these are kind of the outputs that we get from our 5 different assays. As I say, this is the sort of data that we're having to work with. And Tom, who's the scientist who's done most of this work, worked out that he generates per patient sample about 1.25 million data points. So his spreadsheet's pretty big because it goes kind of 1 million across to the right-hand side. And then for each patient, he has that level of detail. But it means we can analyze it in all sorts of ways and look at all sorts of things, including differences between individual cells within the same tumor. This database is huge now, and it's still going to be mined. We're going to use it for a long time to get a lot more data out of it. But what I'm going to show you now is at least the first set of results that we've had from that. So we used about 62 patients in total to go through the process that I've talked about. And what I'm going to show you here is the last 17, where we had a full data set and we used exactly the same assays on all of them. So some of the earlier ones we used for pilot studies. This is the final 17. Each patient is arrayed vertically as we build this graph up. And so you've got 17 boxes across there. And this is the results of the homologous recombination assay. So we were able to define about half as being defective and half as being competent, which funnily enough is exactly what we were expecting. Remember that pie chart I showed you earlier on? We were expecting about 50-50, and that's exactly what we got. So that was quite reassuring, okay? What was maybe a bit disappointing, but perhaps not very expected, was that we weren't able to link that with how these tumours responded to chemotherapy. So as well as testing them for their 5 pathways, we also took a dish of cells and we put carboplatin onto them in the lab to try and see whether the carboplatin killed those particular cells or not. And what we didn't see was any relationship between homologous recombination and whether they responded or not to their chemotherapy. So clearly you need more information than just homologous recombination alone. So then we added in the results of the non-homologous end joining, that's the other double-strand pathway. And I think this is where things start to get quite interesting. So you'll see, again, it's roughly, not quite, but it's roughly half and half, competent or defective. The defective ones are all in purple. And I think what you can see is that there's a bit of mutual exclusivity here. So tumours are either competent in one or competent in the other, okay? Or defective in one and defective in the other. Then we added in the 3 single-strand pathways, and the whole picture now starts to look a bit of a mess, really. It's quite pretty, but I think it's a little bit of a mess. I think the first thing we can say from this is there are essentially no tumours that are exactly the same, okay? They all look really different, okay? The second thing I think we can say is that with the exception possibly of this one, all of them have at least one of those pathways broken. So that really confirmed what we thought we were going to see, that to develop as a cancer, you have to have a defect in one of those 5 pathways at least, okay? I think, with the eye of faith, you could say that the ones here on the left-hand side have more pathways broken than the ones over there, okay? But it's really difficult, and we can't do it. If anybody wants to try, please feel free, but I can't pick out a pattern there that links any single pattern with whether they responded or not to their chemotherapy in a dish, okay? So at that point, we had to use some fairly complex mathematics. Again, luckily, Tom's quite good at this, much better than me, so he did it all. And what he did was he did something called a discriminant analysis, which is where you essentially use artificial intelligence to try and find the patterns in that data that you can't see with your naked eye. And what he was able to do was to develop a pattern or a signature that does define whether the tumour responds or it doesn't respond to chemotherapy. So he can now pick out from this pattern or this result and predict whether an individual tumour is going to respond or not. But of course, that's responding in a dish, and that's very different from did the patient respond or not. So in the next bit, he then linked that with the patient outcome. Now, with credit to my clinical colleagues at St Mary's, out of the 17 patients that we had in here, don't take this the wrong way, unfortunately, they all did really well. So most of them did really well, and only 3 patients relapsed in the space of the 15 months that we followed them up, which is probably slightly better than we were expecting. And that's good from the patient perspective, but it does mean we're a bit limited in trying to analyse this. 1 patient died, but nevertheless, using the same algorithm that he'd developed, the same machine learning tool, he was able to predict exactly whether patients were going to survive or not, or whether they were going to relapse or not in the time period that we showed. So here we seem to have a test that is beginning to work really well to predict whether patients are actually going to respond to their chemotherapy or not. Just to give you an idea, this is the sort of mathematical modelling that he's been using. This is an artificial neural network where you put in all the data on the left-hand side and the computer sort of works out how to model it to come out with the final answer. So it's really difficult to tell exactly which bit of the data is important. We can do that, and we have done that to a degree, trying to tease out our understanding of exactly which bits of all of this work are the really important bits. And we can produce really nice diagrams like that. Okay, but essentially, I think what this work has shown us is that the DNA damage response is complex, which is what we were expecting to see. There isn't a single pattern here that makes easy to understand sense, but we can use it to predict response to chemotherapy. The problem we have at the moment is that they're still in relatively small numbers. So since we did the work that Target funded, we have been able to get some more funding, and we've now run this through a panel of patients who've got relapsed disease. So all of the patients that we've used so far were at the beginning of their treatment course, but we've now done it in another 12 patients who've had relapsed disease. And those data are finished, they're ready, but they're not completely analysed yet. So unfortunately, I haven't got those yet, but so far it's looking like it should work in that group as well. And we think this has got some clinical utility. It's very complicated to set it up. It's been very complicated to get it to work properly. But now that it's working, we can do this actually within about 96 hours of receiving a sample. So if we have a piece of live tumour, so that's the one big challenge, it has to come from the operating theatre straight to our lab. But if you give us a piece of tumour, we can do that in about 96 hours. We can do it with about 2 million cells, which sounds a lot, but equates to about kind of an almond-sized piece of tumour, okay, which is actually reasonably achievable for most patients at the time of surgery. We can give this. And what we've also been able to show is that even if somebody else does it, not just Tom, other people in the lab can do it and they can get the same results. So we seem to have a test here which we now think has clinical utility. It's showing real promise, and we're now testing it in other sample sets to see how we can get on. So it has been a huge success, this project, and I think it's led us to a really good place where we can now begin to think, can we predict response to treatment for patients at an individual level and help them make decisions about which treatment is best for them? Which perhaps leads me on then into the last couple of minutes to just think about where we might be going in the future. So I started this talk showing you that very depressing graph, I think, up in the top right that shows these differences in treatment patterns across the UK. This is something that's been taxing our brains for a long time, and thinking about how we can improve that. And the strategy that we've come up with that we think will hopefully work is if we could use some of the skills and techniques and technologies that we've just shown you in the last few minutes to try and model what would happen to individual patients. Can we help patients to make the best decision for them? So if we could provide really robust and accurate predictions of what's going to happen for individual patients if they have surgery or don't have surgery, if they have chemotherapy or not have chemotherapy, and allow them to try and help them make the best decision for them, would this actually help patients drive some of the changes that we need to see by actually helping them to almost demand what the best treatment for them is going to be? So provide tools that will help accurately predict outcomes. So what we've been doing is, as well as collecting all this biological data that I've shown you, we've been collecting huge amounts of very basic, really, clinical data about all of our patients. And we've now begun to expand that, and we're now including data from 6 centres across the UK. So we're building up a very, very detailed database. To give you some idea, we've got about 2,000 patients registered in that database now, and we have 250 data items for each patient. So age, where they live, what treatments they've had, all sorts of things about them and about their tumour, some of their CT findings, their CA125 results, all sorts of things about them. So 250 things. And at the moment, we're using some of those artificial intelligence machine learning tools that I've referred to earlier to see if we can analyse that and pick out which patients are going to do best with which individual treatments and help them make their own decisions. Ultimately, we would want to try and link that with the sort of biological data and probably even the genome data that we're going to accumulate over the years to generate these really accurate data sets. For anybody who's sceptical about how this might work, well, it's already working because Amazon uses exactly the same technology. So when you go onto Amazon, it will tell you, if you like this book, you're going to like this book. How does it choose that book? Well, essentially what it does is it looks in its database and it finds people like you, and you'd be amazed how much Amazon knows about you, but it knows a lot, and it chooses the books that people like you have chosen. Well, can we use the same sort of technology? Can we say, faced with a particular patient in front of us and all her individual characteristics, can we look in a big database and find patients like her, okay, who are the same age, who live in the same geographical location, have the same BMI, have the same other illnesses in the past, and can we see what's happened to them when they've had particular treatments and use that to generate the accurate predictions? So we're just stealing from Amazon, really. We think this has huge capability for the future. The other thing that's very much on the horizon that a lot of people are talking about, and a lot of people are getting very interested in, is immunotherapy. So immunotherapy is the concept of using the body to fight back against cancer. So we've talked a lot about DNA, and we've talked a lot about the changes that happen in the DNA in a cancer cell. If you have altered DNA, DNA makes protein, so it will make altered and new proteins. So cancer cells express on their surface, they have on their surface proteins that you as an individual patient don't have. They're unique to the cancer because of the changes that are in the DNA underneath it. That means the immune system can recognize those proteins and say, "Oi, this cell isn't a normal cell, it isn't one of our cells," and attack it. And so there's a lot of interest in saying, can we ramp up that immune response and make it work even better? So just to take that further, I said at the beginning, we're all getting DNA damage, and for the most part, we're repairing it. That's one of our mechanisms to stop us getting cancer. The other is we're probably all forming cancers all the time, but for the most part, those are being fought off by the immune system. So again, can we just ramp that up? Key to the immune system and the immune response is the interaction between the T cell and the cancer cell. You have to get a T cell, that's one of your immune cells, to bind to the cancer cell and activate the immune response. A huge amount of work has gone on in trying to understand that interaction. It is really complicated. But what's clear is that it works very well in some cancers, but not in other cancers. And unfortunately, ovarian cancer, it doesn't seem to work very well. Why is that? Well, as usual, it's because it's more complicated. And what we now begin to understand is the T cell and the cancer cell aren't in isolation. They actually exist in what we call the tumor microenvironment. So the area in which the cancer is growing and all the cells around it are all impacting on that immune response. And for some reason that we still don't quite understand, ovarian cancer has a particularly poor tumor microenvironment that doesn't help this immune response. So somehow we need to unpick all of this to understand that, so that we can then supercharge that in the future and try and get that immune response working. And then finally, I said challenge number 3, and the one that we're always talking about is, could we have better screening modalities or early detection modalities? Well, okay, how about this? So I'm going to throw this one at you as an idea, because that's what it is. This is really not much more than a concept at the moment, although I should say there is some really quite exciting preliminary data to say it might just work. And this is work that's coming out of another group in Manchester led by Christine Schmidt that I've been working with for the last couple of years. And Christine and actually her brother Oliver, who's over in Dresden, have been really interested in this idea of microbots or nanobots. And can we make little robots, if you like, to target specific areas? We said right at the beginning that the problem of ovarian cancer probably isn't in the ovary at all, it's actually in the fallopian tube. Can we think of microscopic or nanoparticles, if you like, that might be able to get to the fallopian tube? Well, yes, sperm are ideally suited to get into the fallopian tube. That's what they do. Could we take sperm, open them up, put a chemotherapy drug inside, close them up again, target them against those pre-cancer cells that I showed you in the fallopian tube, set them off on their little journey, get them to bind to those pre-cancer cells, offload their chemotherapy and kill the pre-cancer before it ever gets into being a cancer, okay? I leave it as a concept, all right? I leave it as a concept, all right? I knew this will generate most of the discussion, okay? I know that. But just let me tell you, the bits that we can do already, and when I say we, it's not really me, it's really Christine and her brother. They are able to take sperm, they are able to put the chemotherapy drug in it, they are able to get that to then bind to a cell and offload its chemotherapy into that cell and kill that cell. What we've not been able to do yet, but we're working on, is target that into the fallopian tube cells that we're interested in. And a key bit of what we're trying to do at the moment is find the right binding agent, if you like, the right key and lock system to get the sperm to bind to the cells that we want. So we're still an awful long way away from this being anything of clinical use, but I just leave it to you that understanding that biology and unraveling biology, which is why this sort of research is so important, can lead to these kinds of discoveries and this kind of thought process. Whether this ever comes to fruition, I don't know, but at least there is some hope out there and we are trying. At that point, I'm going to leave it and just say a huge thank you to everybody who's been involved, not least Target Ovarian Cancer for funding this project, but the clinical teams at St Mary's, which is over on the right-hand side, and the sciencey side from the university. And this is just some of the people who've been involved over the years in helping us collect samples, process those samples, do and develop the tests that we've talked about, and lead that through to the work that we've presented to you. I hope I've shown you that this is a technical challenge. It is really difficult stuff to do. It takes a long time to do it, and there have been a lot of false routes along the way to get to where we've got to. So I've presented a very simplified outcome of sort of 4 or 5 years' work, and that's why this sort of research does turn out to be expensive. But at the end of the day, I hope we show you that this type of research really does have benefits and has set us up very well for the future. So I'll leave it there, but very happy to take questions. Thank you.